Sorry, what?
Not full screen yet.
No, it should be.
Now, okay.
Okay, so I may start?
Yeah.
All right. Dear colleagues, I'm very glad to see and hear everyone who came today to hear Evgeny and me present our talk. Today we'll be sharing a part of the results of our project, which we have been working on for the past 2 years, and which we finished at the end of 2025. Our project was called Wolfson Conifers of the Permian and Triassic of Angaraland, Systematics and Phylogeny. The project had 2 goals. The first one was to find and study remains of Wolfson conifers from the Permian and Triassic of Angaraland, particularly Siberia. Here is one moment. Here is a list of some publications related to the first goal and the ultrasynth section of the pollen, which you saw on the banner of this year, is a photo from this publication. It was a pollen extracted from new Borrisyak torbus from Siberia. But today I will speak more about the second goal, which was to attempt to understand the systematics of early conifers. Angaraland conifers are little taken into account in phylogenetic constructions, but we continue to receive new data every year, and of course they should be taken into account. There are currently several points of view on the taxonomy of early conifers. This slide shows only a few of the most widely accepted ones. Current views can be grouped into 2 main concepts. The prevailing concept unites early conifers in a single group, which is ancestral to the living conifers. This concept is also supported by phylogenetic analysis. However, there is an alternative viewpoint, according to which early conifers are divided into 2 groups, the so-called Walchielian and Waltzelian conifers. This view on the taxonomy of early conifers was proposed by Valentin Krasilov in 1971, but a similar idea was later expressed by Sergei Mein in 1997. Krasilov proposed to recognize the independent order Walchialis with Waltzelian conifers as basal representatives of the order Coniferalis. Mein distinguished the families Libachelaceae, which corresponds to the Walchielian conifers, and Waltzaceae. Both Krasilov and Mein believed that Walchielian and Waltzelian conifers should be separated based on the structure of their female reproductive structures. In Walchielians, they are represented by fertile shoots with sterile scales and seed stalks, while in Waltzelians, fertile shoots are reduced and transformed into a seed scale. All previous phylogenetic trees showed that the Walchielian and Waltzelian conifers form a monophyletic group. Gary Rothwell, Jean Mapes, and Gennaro Hernandez Castillo found Walchielian conifers to be ancestral to Waltzelians. If Walchielians were separated from Waltzelians, they would form a paraphyletic group. Fabiani Herrera and co-authors modified the previous character matrix and analyzed a larger number of taxa, but their resultant picture remained fundamentally unchanged. However, previous constructions did not include early conifers from Angaraland described in recent decades. We were curious to see whether the results of the phylogenetic analysis would change if we added Angaraland taxa. The taxa we have chosen are Archaeovolce, Sashinia, and Mutovia spermum. They represent early stages in the development of early conifers on the East European platform during the Permian. Below, I'll discuss each of them in more detail, but first I'd like to briefly discuss the terminology I'll be using. I will use such schemes for the terminology unification. They represent a simplified structure of a female fructification in different genera, which do not represent all details like a paleobotanical drawing or a photo. So, going back to the terminology. The axillary Brecht complex, also known as the Brecht scale complex, includes a Brecht, a modified leaf, and a fertile shoot, which is a female reproductive structure that develops in the axil of the Brecht. The fertile shoot is the evolutionary origin and anatomical precursor of the seed scale. I'll refer to the structures in the axillary Brecht complex that bear seeds as seed stalks and the sterile structures as sterile scales. Archaeovolta originates from the lower and middle permen of the Caesurals. These plants had fertile zones on the shoot rather than cones. The fertile zones contained axillary Brecht complexes consisting of a seed scale and a Brecht. The seed scale bore 3 to 6 seed scars. The Brecht was long and linear, and the leaves of these plants were linear with a single vein. It is suggested that plants with female fructifications of Archaeovolta may also have male fructifications of genus Uralostrobus. Uralostrobus is reconstructed as a cone with peltate microsporphyls. The sporangia were located on the inner surface of the shield on the underside, and structurally, Uralostrobus was very similar to Wilsiastrobus. Sachinias were an important component of the middle and late Permian vegetation on the East European platform. We included Sachinia antiqua from the middle Permian in our analysis. These plants had unreduced fertile shoots with well-developed seed stalks, but without sterile scales. These fertile shoots were underlain by a Brecht, which was modified compared to the vegetative leaves. Sachinia antiqua is associated with shoots of Quadracladus antiquus and male fructifications Cargalostrobus dimetri. Cargalostrobus have peltate microsporphyls with a shield and a stalk, and the sporangia are attached to the stalk. Plutoria is known from Upper Permian deposits of the East European platform. It is a rather unusual and enigmatic plant interpreted as a conifer. Its female fructifications are apparently represented as an Archaeovolta by fertile zones on leafy shoots. The fertile shoots are reduced to some degree. They become peltate with seeds on the lower side of the disc. The individual lobes of the disc appear to be flattened and partially fused seed stalks. Sterile scales are absent on the fertile shoot, at least on the stalk and the lower side of the disc. Shoots of Quadracladus schweizeri have been found in organic connection with Matoia spermum. These female fructifications are also associated with male fructifications of Dwinostrobus. Their microsporphyls consist of a stalk and a shield, but they are non-peltate, and the sporangia are located in pairs on short sporangiophores on the stalk. So let's move on to the results. We added these 3 taxa to the character matrix we borrowed from Herrera and Coto's paper published in 2020. Their matrix is based on Rothwell, Mapes, and Hernandez Castillo's matrix published in 2005, but was significantly revised. We made no changes to the original matrix. Thus, our analysis included 42 taxa and 52 characters. This is the consensus tree we obtained. The consensus tree generalizes several trees and displays only those clades that are found in the majority of the original trees. Our tree shows orders Dicronophyllalis, Rogliocladalis, Venovscialis, Cordaetantalis, and Callistophytalis. But more interestingly, the early conifers in our tree split into two clades. Moreover, the majority rule consensus tree shows a 70% support for the divergence of these two groups. And these two groups of early conifers correspond to the Walchelian and Waltzelian conifers as was defined by Krasilov. We looked at which characters and how they are distributed across the tree. It turned out that the features distinguishing Walchelians from Waltzelians are similar to those used by Krasilov and Mann to separate these two groups. Here are a few interesting features. Firstly, there are a couple of characters common to almost all early conifers. They relate to the structure of fertile shoots. Most early conifers are characterized by bilateral symmetry of the fertile shoot. Another character typical for almost all early conifers is inverted seeds. But significant differences between Walchelians and Waltzelians are observed beyond that. First, in Walchelians, the seeds are placed on developed seed stalks, while in Waltzelians, the seed stalks are fused to a lesser or greater degree in a seed scale. Second, in Walchelians, the seeds are located near the edge of the seed stalks, roughly speaking, in the terminal part of the fertile shoot. In Waltzelians, however, the seeds are shifted towards the middle of the seed scale, and since the latter is homologous to the fertile shoot, it can be said that the seeds are shifted towards the middle of the fertile shoot. In some Waltzelians, they are even shifted towards the base of the seed scale. Thirdly, Walchelians are characterized by the presence of a large number of sterile scales on the fertile shoots, more than 6 or even more than 20. Waltzelians typically have no sterile scales, but there is a clade in which sterile scales have either been retained in small numbers or have evolved anew. There are also some interesting characters related to bract structure. The first is whether the bract is modified compared to the vegetative leaves. As you can see in Waltzelians, it is almost always modified, while Walchelians initially typically have unmodified bracts, but then a group appears in which the bracts are also modified. The second character is the presence of a forked apex of bract. This feature is not found in Waltzelians, but in Walchelians there is a clear tendency for bracts to be forked. The third character is the presence of trichomes on the edge of the bract. Waltzelians, for which this character is established, lack trichomes, but most Walchelians do have them. The male fructifications of early conifers maybe have been studied as much as the female ones, but unfortunately, establishing the relationship between male and female fructifications is not always possible. For many taxa in our analysis, unfortunately, information on the structure of male fructifications and pollen is lacking. But if we look at the distribution of traits across known taxa, we can also observe some interesting things. For example, Walchelians are characterized by pre-pollen with a proximal scar. As can be seen, pollen with a distal aperture is more typical for Waltzelians, while pre-pollen with a proximal scar is known in only one case. Of course, this image first of all illustrates the need for further study and whole plant reconstructions, but even the available data reveals a curious pattern. Thus, it is possible to identify complexes of characters that allow us to distinguish between the Valhalians and Valcelians. Valhalians are characterized by fertile shoots with sterile scales and seed stalks with subapical seeds. Bracts of Valhalians tend to be forked. This group is characterized by pre-pollen with a proximal scar only. Families Tucidiacea, Empyreacea, Valhiacea, Cumgorodendracea, and probably Scienciacea belong to Valhalians. Valhalians are distinguished by reduced fertile shoots lacking sterile scales. Their seed stalks fuse to a lesser or greater degree into a seed scale, and the seeds are displaced lower towards the base of the shoot. The bracts are usually modified by comparison to vegetative leaves and remain unforked. Valhalians possess pollen with a distal aperture, while the proximal scar is often lacking. Families Patychiacea, Maniniacea, Valiciacea, and Crassiloviacea belong to this group, as well as probably Conhalepis. We also analyzed our tree from a stratigraphic perspective and obtained the following picture. The groups we identified are quite consistent with the stratigraphic distribution of taxa. Our diagram suggests that Valhalians arose as a result of the first stage of intensive diversification of early conifers in the late Carboniferous. Valcelians arose as a result of the second stage of diversification in the early Permian and underwent another stage of diversification in the Middle Triassic. Of course, our tree isn't without its flaws. In particular, the placement of the genera Conhalepis and Sashinia raises some serious questions. Conhalepis is placed at the base of the branch leading to Valhialis and Dicranophyllalis, although its structure is quite comparable to that of Volcelians. Conhalepis has flattened fertile shoots, reduced seed stalks, and seeds slightly displaced toward the middle part of the shoot. However, the fertile shoot is covered with outgrowths whose homology is not yet entirely clear. This could be sterile scales, sterilized seed stalks, secondary outgrowths, or something else. Most likely, Conhalepis was placed in this position precisely because of problems interpreting its structure. In case of Sashinia, there are no problems with interpretation. Their fertile shoots are structurally consistent with those of Valhalians, yet in our strict consensus tree, they form a clade independent of both Valhalians and Valcelians, which is rather strange. In our majority rule consensus tree, Sashinians are closer to Valcelians, but this dichotomy has the lowest support in the whole tree. We suspect this is due to the fact that we selected an unrepresentative species. Sashinians are generally interesting plants because their older representatives show more advanced characters than their younger representatives. Sashinia antiqua, which we used for the analysis, is a middle Permian species. It has reduced sterile scales on the fertile shoots, and the associated male fructifications have peltate microsporophylls, which is characteristic of the Volcelians. Late Permian Sashinians, however, appear more primitive. They retain sterile scales, and the associated male fructifications, which belong to a different genus, have non-peltate microsporophylls. The problematic position of Sashinia also raises questions about the position of Motovia spermum. At first glance, Motovia spermum doesn't look very similar to Sashinia. but the fertile shoots of Motavaspermum can easily be deduced from fertile shoots of Sarshenia by imagining that the seed stalks placed at the shoot apex are partially fused and the lower seed stalks were reduced. Moreover, both genera are associated with the vegetative shoots of Dracladus and male fructifications of Dinostrobus type. However, Sarshenia and Motavaspermum are placed quite far apart on the tree. Another disappointing aspect of our results is the bootstrap analysis. The supports I mentioned earlier were obtained using the majority rule analysis. We also conducted bootstrap analysis, but unfortunately, its results yielded polytomies for almost all taxa. This means we still have a lot of work to do. During our work, we also noted some methodological aspects in the Herrera's matrix that, in our opinion, require refinement. First, there is ambiguity in terminology. For example, there are a number of characters describing the structure of the leaf or bract subtending ovule or ovulate cone. Rothwell and Cotters had a more appropriate term for this category of characters, for example, leaf or bract subtending ultimate ovule-bearing structure, meaning the bract of the axillary bract complex. In Herrera and Cotters formulation, this character can also describe the bracts underlying the entire female fructification or a sterile scale subtending the seed stalk. Another example can be found in character 47, distant stalk on ovuliferous shoot or ovuliferous scale. In practice, it turns out that this feature was used only in seed scales with a stalk, although formally many Wilhellians with a developed fertile shoot also have a developed stalk. Secondly, there is the repetitiveness of certain characters. This category includes characters related, for example, to the structure of sterile scales on the fertile shoot. These are number of sterile vegetative scales on ovuliferous shoot, integrating vegetative and fertile scales, and vegetative scales on adaxial and abaxial surface of ovuliferous shoot. These characters could serve well to distinguish groups, for example, within Wilhellians, but in practice, it turns out that all of them point to the same thing. If a conifer has sterile scales on the fertile shoot, then they are present on both the abaxial and adaxial sides, and they transition into the seed stalks in one way or another. However, 4 characters are devoted to this characteristic, which may artificially inflate the significance of the sterile scale structure in phylogenetic analysis. Perhaps this is why Sarshenia antiqua is closely related to Wiltzellians, as it, like Wiltzellians, lacks sterile scales. Third problem is the presence of internally non-homologous traits, for example, character 40, position of ovules on sporophyll or fertile scale or ovuliferous scale. In this case, it's quite clear that the seed scales and sporophylls, in our terminology, seed stalks, are non-homologous, yet they are considered together in this character. And fourth, of course, is a problem with whole plant reconstructions. The tree is constructed primarily based on the structure of female fructifications. Next come traits devoted to the structure of vegetative structures. But as is well known, the vegetative structures of conifers are highly polymorphic and prone to parallelism, making them less informative. And third place is occupied by the structure of male fructifications. For many taxa, information on their male fructifications is lacking, although it would undoubtedly be very valuable. So let's summarize. Previous attempts to conduct a phylogenetic analysis of early conifers showed that they form a monophyletic group. We added only 3 new taxa of permanent conifers from Angaraland to the matrix, and our analysis showed that early conifers fall into 2 monophyletic groups, corresponding to Walchelians and Valsalians as defined by Krasилov. Moreover, Walchelians arose as a result of the first stage of intensive diversification of early conifers in the late Carboniferous. Valsalians arose as a result of the second stage of diversification in the early Permian and underwent another stage of diversification in the Middle Triassic. Our data are still preliminary and require further refinement. We plan to revise the character matrix of Herrera and Quarters, addressing issues to ambiguous terminology, repeating characters, and non-homologous characters. We will also add new taxa, in particular the late Permian Sachinias and other recently published Valsalians from Triassic and Permian deposits. Thank you for your attention.
Thank you, Tatiana, for so informative and interesting talk. And now time to questions. Susan Renner, you may switch on your... Yeah. Welcome.
Thank you. I was just applauding, but since I'm on now, I do want to ask a question. Among the methodological problems, I see the application of cladistics, because if there are surviving ancestors in this data set, which is very plausible, then, as you know, they will invariably result in polytomies, in unresolvable relationships. And therefore, it might be better to apply a method that does not assume purely sister group relationships, such as network analysis or other approaches.
Evgeny, I think you should join us in this talk because with methodological aspects of cladistics, I'm afraid to say something wrong because Evgeny knows this area much better than me.
About methodology, use another method, not...
The assumption, the underlying assumption is that everything can be depicted by dichotomous relationships. And if there's a polytomy, we need more characters. But the fact is that if there are surviving ancestor and descendant relationships in a matrix, then there will be invariably polytomies that cannot be resolved, no matter how many characters. And this is not an opinion, this is mathematically clear. I really, really understand the problem. I loved your talk, okay? The problem is that we do not know who may be the ancestors and who may be the descendants. But pure likelihood tells us that some of these fossils are in fact ancestors of other lineages that survived that are also in the matrix. That is a plausibility argument. It's not at all a question about the validity of your analysis. It is a question about whether the application of cladistics presents a methodological problem that also contributes to the unresolvable relationships that you have shown us. In your tree, you know, the strict consensus tree and the bootstrap tree. They are not only unresolved because of lacking characters or possibly miscoded characters, they are also unresolved because there are ancestors and descendants in the matrix. That's all I want to say. I should stop. Thank you again.
Thank you very much. It's really very, very useful because we just start in this field of science, and I think we are happy for any advice.
Thank you. More questions?
Okay, may I ask?
Who is speaking? Alexei?
Dmitry.
It is me.
Dmitry, okay. Welcome.
First of all, it's fascinating. It's fantastic to attend this talk. And I just have a question. If the two groups
are both monophyletic, and one group is much younger than the other group, then we assume that there was a ghost lineage of the younger group. So what can you say about possible history of that ghost lineage? What happened to them during this period of time? To the stem group, to the ancestors during this period of time?
Very good question. Thank you. What could happen to them? Well, actually,
we should assume that there were some fossils representing that lineage, yes, because if they appeared much later, then something should happen to them, to the ancestors.
Well, actually, it's not so much later. There's a picture.
Yeah.
Because they, I think this problem maybe is connected to what Suzanne was explaining to us about ancestor and descendant relationship. I'm not sure, of course, but maybe there was a group ancestral for both Valhalians and Valsalians, and maybe some representatives of this group we identify as Valhalians, maybe, I'm not sure.
Mitya?
Thank you very much. Yes, this kind of question shouldn't have a good answer. I'm sorry about that.
That's what we.
Thank you anyway.
So no evidence of latest Wilhelms in before after Permian-Triassic boundary. We have no conifers like Wilhelms, basically.
Thank you. Next question. I do not see hands, but you may write in the chat if you prefer. May I ask one, Tatiana or Evgeny, how many new taxa are needed that the tree remains the same?
It's also a good question.
Without good answer.
Good.
You have added 3 taxa, if I am not mistaken, and so the picture, the resulting picture is different. And if you add 3 more taxa, for example, will picture be the same or a new one?
We will not know it until we add new taxa, I suppose.
Okay, thank you. And I see something in the chat. You may also look in it from Susan again. So are the taxation not part of the clade formed by the other surviving conifers?
Taxation, I need to take a look. Так. Can you repeat the question, please?
Look in the chat. So are the taxation not part of the clade formed by the other surviving conifers?
This is because you showed us a slide in the beginning that was the Krasilov hypothesis. And I made a screenshot because I found it so interesting to really see this explicitly stated that taxaceae derive. Yeah, thank you.
Yes, man supposed.
It was the Mayans hypothesis, excuse
Yeah,
me.
yes, it was. It's the man supposed. Krasil supposed that all living conifers
sorry.
originated from Precedians.
A question, look also in the chat, from Bruce Cornett. Are there examples of conifers in Eurasia where ovules are enclosed in bracts, such as in Araucaria?
In Eurasia,
I don't know such. Evgeny Vladimirovich, вы знаете?
I think enclosed in brick. No, I had no such question.
Don't know the example.
Conifers, I don't know. Araucaria.
Okay. A remark from Evgenia. Thank you for your talk. I do not see hands. So, and I do not see.
I can ask more questions if you.
Yeah.
If you have.
You're welcome.
So, I was just recently I was recently trying to read some literature about those questions, and I found there is, for example, such a family, Paleontological. Yes. So where is it in the phylogeny? Or even maybe simpler question. There is a family Hierolipidaceae. Okay, if my pronunciation is.
Hierolipidaceae.
Yes, it is a
probably
large, with Classopolis pollen. Yes. So they should be somewhere. Okay.
Yes. Hierolipidaceae, as I remember, they are. It is a younger conifer.
It is. Yes, it is a younger. Of course, it's
and
younger.
We
But
didn't include it in our analysis. Maybe we should have, but we didn't. These are mostly Permian and Triassic conifers, and just a few modern conifers.
Yes, but yeah, I understand. This I understand. But if you are interested in the question whether, for example, Voltzialian conifers Voltzialians are monophyletic, okay, already from your tree we see that with respect to Pinales, your Voltzialians are not monophyletic.
Yes, they are not monophyletic. It was incorrection.
Yes. But so the main and very much fascinating conclusion of your analysis is the proposed monophyletic nature of Valhalians. This is very novel, yes? And this is very interesting.
Yes, it's very interesting. I suppose that Hierolipidaceae should be somewhere within this clade.
Okay.
Try to include this to analysis. And
I'd like to remark, as I remember the paper about this plant, they are very unsure about the position of the family, and they relate it with Valtzialian in I don't know what sense. And it is quite different from Hierolipidaceae. So yes, it's interesting to think how it would be related or not related with the families the colleagues are comparing in their analysis.
So I think adding younger groups will be. I think adding younger groups will be really important for the analysis. In my view,
of course.
Sure. Okay. Well, I'll read the next comment. Bruce Cornett writes that Nevark supergroup of Northeast America contains many taxa that produce Classopolis or Araucarian pollen. All this relate to later times, I suppose. Well, I'm looking for more questions or comments or answers to previous questions.
I have a small comment about this. So we do not believe that cladistic is the best method for using for reconstruction of systematics of fossil groups and fossil systematics. But we try to follow previous studies of conifers after Rothwell, Herrera, and try to obtain new results, and we show you.
Thank you, Evgeny. And
may
I want to
ask
answer. Now that you brought this up, I want to say something. I completely understand I really understand the problem. Okay,
Yeah.
but we could use multiple methods. How about using two methods and then comparing them, or presenting in the online supporting material an alternative analysis with network analysis, which is done in seconds, as you know. You upload the matrix, you do a network, and you compare. Or other visualization approaches. I think that would be useful, might be useful. That's all I want to say. I'm not against cladistics, okay?
Yeah. Thank you.
it seems a good advice for further work.
May I ask one more
Yeah,
question?
surely.
Yes. So there is Dicronophyllum, and as far as I understand, there were at least two ideas about relationships of this fossil. Some people suggested that it is related to early Gingoalian fossils. Yes.
Yes.
So what is your opinion about this idea? So you are sure that it is related to conifers? Yes, broadly speaking.
Evgeny?
No, Dicronophyllum is, of course, we represent as a primitive group of, and
but conifers or gingos?
That's interesting, and maybe also with Tejdesperms, maybe also correlate.
Mm-hmm.
No.
Mm-hmm.
What we also include as conifers, such as in previous matrices of Herrera and Rothwell, whereas the Dicronophyllum we include in this analysis.
No, I mean, I'm asking about your personal opinion as a paleobotanist and as a great expert in fossils. I'm not asking about cladistic analysis, just your personal opinion. What do you think personally about Dicronophyllum?
I think it is maybe related with conifers such as Weinovsky, for example, plants, the enigmatic plants. And uncertainly, the groups may be more closer to conifers or closer to gingos. I don't know.
Okay, thank you. Thank you very much.
Thank you. Well, more questions or comments?
Can I ask some questions?
Yeah, Alexei.
I have just now look in detail for this shim, for this cladistic tree, and I have some questions about it. For the first, where is Mayonica in this tree? And the whole family of Myonicaceae.
Myonica is here.
Oh, I see, yes. And the second, why do you include only Sascinia antiqua and don't include Sascinia aristavensis and Sascinia borealis?
We understood that it was a mistake after we got the tree. Well, Sascinia antiqua was more ancient.
Alexei?
I think that the age of
Yes.
taxa should not be.
I think we add the Sascinia aristavensis later and try to again this analysis. I'm not sure why
So
we
these are plans for the future
yeah
to add all species of this genus.
yeah
Because yet
with the genus
it's a very
Trombus.
problematic position.
Yeah.
Maybe then the position of Sascinia antiqua will be another.
Maybe I hope for this, because yet it has a very little sense.
Alexei, no comments?
Yes, no.
Okay, I see only, I see one comment from Bruce related to the one of the previous questions that it was about Jurassic, Classopolis and Araucarian pollen, and Bruce also shared a paper. Anybody who wished to read it can download it as long as we're here in the conference. Well, and I see a message from Elena Burkanova, Tatiana, and Evgeny. Thank you for your talk. Well, do we have more questions, comments? I see. Wow, one more comment from Bruce that the Herolipid conifer produced. Oh no, sorry. A question from Alexei Askolsky. Thank you so much for the excited talk. Are there any data on wood structure of Valhalis?
Yeah, in our matrix, in matrix data about wood, absolutely absent. And not only one chapters in our matrix about wood structure conifers.
No data.
Yeah. Not included. And not summarized data.
Haru Nishida writes, thank you for nice talk. That leads me to think again about conifer history, and congratulations to the 5 years achievement of the seminar. Bruce also writes, I did not know so many conifer cones have been studied. Thank you. More comments or questions or answers or advice?
Okay.
Well, it seems that we have finished. Thank you very much for coming, for joining us, and come again.
Thank you very much.
Bye.
Very interesting.